Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. So today we will discuss about the DNA structure. So let's talk about the DNA structure. So here is the multiple cell, which that is the cell contain a nucleus, and each nucleus contain a DNA set of the chromosome, which is the dense formation of the DNA. So as the chromosome contain a chromatin as a DNA, and centromere are present, which that is bind to each other. And so this is the thread-like structure is bound with the histone protein. So this histone protein is a 4 unit, and these histone proteins tightly bound, as well as the linker protein is helper protein. While this uncoiling of the DNA due to the methylation or acetylation. So the acetylation is the unwinding of the DNA, while the methylation group which that is attached, that is known as the molecular tag, which that is suppressed, while the acetylation acetyl group is expressed. So that is here is the DNA. DNA contain a two strand. So the DNA is the basically deoxyribonucleic acid. So this is known as the DNA. DNA is a double strand, which that is a helical structure, and that is made up of nitrogenous base and phosphate group as well as sugar. So the deoxyribonucleic acid or DNA genetic material. So how it's produced as a structural protein whole body, which that is produced as protein, is the in this manner we will discuss here. So the general dogma of the DNA expression in the form of phenotype from the genotype is here. That is the transcription in the nucleus to produce messenger RNA, for example. So these messenger RNA contain a ribose nucleic acid. So that is the nucleotide subunit is made up of RNA. So that's RNA polymerase is used to produce the RNA from the DNA. So this is the ribonucleic acid or RNA is the genetic material, is come from the genetic material, and that's after the translation in the cytoplasm of the cell to produce protein, and that protein is the unique for the unique work for the structural and functional protein is used to produce, for example, the antibody production or muscle protein or something, you can say everything. So here is the composition of DNA we will discuss here. That is, here is the DNA composition of the nitrogenous base is the purine. Purine is the double ring, is known as the adenine either or guanine. So the pyrimidine is a single ring. So the single ring contain a cytosine or thymine, while in the RNA contain uracil also. So the four nitrogenous bases are present in the DNA, while the uracil is also present in the basically in the RNA instead of the thymine. So here is the basically one nucleotide which contain a ribose sugar as well as nitrogen base and phosphate group is attached carbon number 5, and the second one is attached with the carbon number 3 of the sugar. So the deoxy adenosine 5 monophosphate is known as the DAMP. So I hope you make sense about the general structure of the DNA. Now we will discuss about the Watson and Crick model of DNA. So the Watson and Crick model of DNA is most explanatory power of the DNA, and that discovered in 1950 and covalently so discuss here. So the covalently bonds and hydrogen bonds are present. So what is the meaning of this? Next we will discuss also, but here we will just understand that is the DNA is helical in the shape. So the helical structure is formed in the DNA due to the coiling. So So the two strands are present. That two strand is made up of phosphate group and sugar ribose. Let's here I am drawing the DNA structure is the backbone of the DNA, which that contain a phosphate group as well as ribose sugar. So here is the phosphate group are attached and deoxyribose sugar are present in the form of DNA, while the oxyribose sugar are present in the RNA. While here we will discuss just and understanding the Watson and Crick model of DNA. So that phosphate basically here I am drawing that is the two strands, so that the helix has a uniform width and of 2 nanometer. So their width is the 2 nanometer. We will discuss in later. So here is the nitrogenous base are 0.34 nanometer apart. It means the two nitrogenous base which that are ally basically one nitrogen base to the another nitrogen base is the gap which that is covered by the 0.34 nanometer gap. So here is the DNA structure which that is showing in the figure, which that animation is looking like this. So that is here is the minor groove, while other is the major groove which that is showing in the figure. So that is a minor groove is looking like this, and that's basically, and major groove is here. So between the minor groove and major groove, that is the distance, and this distance is one turn, and this one turn is made up of length is 3.4 nanometer. So this 3.4 nanometer is the length from the one turn of the DNA double stranded, while the between the two nitrogenous base is here. For example, the one pair of the nitrogenous base by the hydrogen bonding, after the next is the distance is 0.34 nanometer. It is also known as a 3.4 angstrom. So I hope you make sense about this concept. So let's talk about again Watson and Crick model of further point. So here is the just width is 2 nanometer, so the diameter of 20 angstrom, and this diameter is the DNA diameter, which that is coiling like here. So here is we will discuss about the 10 base pair are present on each turn. It means the 10 base pair are present in each turn of the helix. So the next is the phosphate group for present outside the helix, but the nitrogenous base are present in the interior of the double helix. So the inside of the double helix, that is nitrogen base, are present, and both nitrogen base one strand and second strand to attach by the hydrogen bonding. So remember, so the double bond, so the side ropes are the equivalent sugar phosphate backbone. So the here is the nitrogenous, basically here is the nitrogenous base in the form of pair. So this pair formation. So Franklin's X-ray data shows that indicate that helix makes on full turn after every 3.4 nanometer of its length. So there's length is turn one helix. So that's the pairing of the nitrogenous base is complement. It means the complement. What is the meaning? For example, we will discuss here. Complement means the if thymine are present and the adenine is unfortunately will be occur. It means the thymine is attached only adenine by the triple bond of hydrogen bonding. On the other hand, the thymine also, but cytosine attached with the guanine is the triple bond, while adenine thymine is attached here, that is the basically, and each strand is the anti-parallel. For example, one is going to 5 prime to 3 prime phosphate group attached with a ribose sugar, while the other is the opposite. So here is the triple bond is present in the thymine to adenine formation, while the cytosine to guanine is a double bond. So that is the indication for it. while here is you can see that there is this little bit my mistake, so you can correct. It means the cytosine attached with the guanine with a double bond, while the thymine is attached with the adenine. Adenine is a conformation, and that is the triple bond. So the triple bond is the thymine to adenine, while the cytosine to guanine is the double bond. So I hope you make sense about that. It means here is a nitrogenous base, which that is basically nitrogenous base in the form of pair, and that's Franklin and X-ray. Franklin's X-ray data indicate that helix makes on full turn after every 3.4 nanometer of its length. So the pairing of the nitrogen base is complement. Complement mean if adenine is present, so the other is opposite nitrogen base will be thymine and not other guanine or something. So here is we will discuss the pairing of the nitrogen base is complement. So both strands are anti-parallel, parallel. Anti mean against. So that means the parallel. So the parallel mean both are equal, but against. For example, 5 prime to 3 prime. So other is the 3 prime to 5 prime. So here I am completed. I have completed this topic about the DNA structure. Next we will discuss about the DNA replication in eukaryotes. So that will be next video, but here I will draw the hiding to get the next video to pick up the DNA replication, so you can understand also the DNA replication. And next video we will also understand about the transcription and translation of the DNA with explanation. So